Good afternoon, students. Welcome back to zoology class. In yesterday's class, we have discussed about first line of defense, second line of defense, and third line of defense. See, we have two types of immune systems, that is non-specific immunity and specific immunity. Non-specific immunity is the generalized response of our immune system to the pathogen. Whereas specific immunity, as the name indicates, it is specific response of our immune system to the particular pathogen. The non-specific immunity contains first line of defense, that is physical and physiological barriers. Physical barriers contain skin and mucosal tissue, whereas chemical barriers, as we have discussed, tears, saliva, and pH present in the stomach due to HCl, all these act as chemical barriers or functional barriers or physiological barriers. And in the second line of defense, we will come across with cells of our immune system. The cells, the cellular barriers, white blood cells, macrophages, and interleukins, all these we have studied in the cellular barriers. And next, coming to third line of defense, the actual immune system, which includes T lymphocytes and B lymphocytes. See here, in the third line of defense, we have discussed about T lymphocytes and B lymphocytes. Apart from that, what is antigen and what is antibody? Antigen is a foreign material which is capable of eliciting an immune response. Say for example, a pathogen or any physical agent or any biological agent or chemical agent which is provoking our immune system to fight against with that is known as antigen. Then what are antibodies? Antibodies are the proteins which are produced in response to antigen. Those are known as antibodies. Antibodies. These antibodies are chemically, they are proteins. Okay? And they belong to the group globulin. Globulin proteins. So, the antibodies are also known as immunoglobulins. Immunoglobulins. And we will refer them as Ig. And these are highly specific to antigen. Antibodies are highly specific to antigen. Antibodies are highly specific to antigen. And there are different types of antigen: IgA, IgE, IgM, and IgD. Alpha immunoglobulins, epsilon, mu, and delta. The smallest of all the antibodies is IgG. IgG is the smallest antibody which can pass through the placenta. If you have recollect, in the human reproductive system we have studied that these IgG antibodies can cross the placenta and enter into fetus. Such type of antibodies are known as transplacental antibodies. Transplacental antibodies which can cross the placenta from the mother and they will enter into fetus, and it is the smallest antibody. Do remember? And even the G is the most abundant antibodies. IgG are the most abundant antibodies. And largest antibody, which is a pentamer, it is IgM. It is a pentamer. Okay, remaining. The antibodies which are responsible for allergic reactions are IgE. These can be seen during allergic reactions. In detail, we are going to study about this. So there are 5 different types of antibodies: IgG, IgA, IgE, M, and D antibodies are there. Coming here, this is the structure of antibody. If you have remembered who discovered antibody, antibodies are discovered by Emil von Beckerin. He is the father of immunology. The yesterday's class we have discussed. No? Antibodies were identified by Emil von Beckerin. And if you see the structure of antibody, antibody is a Y-shaped structure. And it is a tetramer. Antibody is a tetramer. Tetramer means having 4 chains. How many chains it is having? 4 chains. 4 chains, 2 chains are heavy chains and 2 chains are light chains. These 2 are heavy chains which are large, red in color, and 2 chains are short and they are known as light chains. And this antibody, it is containing 2 regions. One is constant, this is constant region and the other one is variable region. This region is known as variable region. And whereas this region it is known as constant region. Why it is variable means to these regions antigen can come and bind here. Different type, a particular antigen comes and binds here, so that region is known as variable region, whereas the opposite side it is constant region. And heavy chains and light chains, the 2 heavy chains and even the heavy chain and light chain are joined by a disulfide bond. Disulfide bonds will help in connecting the heavy chains and light chains. As there are 2 heavy chains and 2 light chains, this antibody can be written as H2L2 molecule. H2L2 means what? 2 heavy chains and 2 light chains, which is a tetramer. In the variable side, there is a particular site for binding of the antigen. The site which is helping for binding of the antigen to the antibody is known as paratope. What it is called? Paratope. And the site which is present on the antigen is known as epitope. Say for example, an antigen has comes and it has binded here. Now it can be forming an antigen antibody complex. Antigen antibody complex. The site present on the antigen for binding with the antibody is known as epitope. The site present on the antibody for binding with the antigen is known as paratope. When epitope and paratope combined together, it forms antigen antibody complex. Is that clear? Once again, see the structure of antibody, important even for board exam also. See, structure of antibody, it is a Y-shaped structure. These antibodies were discovered by Emil von Beckerin. Each antibody is a tetramer having 4 chains. Out of those 4 chains, 2 are heavy chains and 2 are light chains. The heavy chain and light chain are connected or joined by disulfide bonds. An antibody consisting of 2 regions, one is variable region where it can bind with the antigen. The opposite end is constant region, which is known as FC region or fragment. FC region means fragment. FC region means crystallization region. Whereas the opposite end is known as Fab region. This Fab region means fragment antigen binding site. Is that clear? This is the structure of antibody. These antibodies are produced by B lymphocytes. T lymphocytes do not have the capacity to produce antibodies, but they will help the B lymphocytes in the production of antibodies. Is that clear? These are proteins which are produced in response to antigen, and they are called immunoglobulins represented by Ig, and they are highly specific in their nature to the particular antigen. There are 5 different types of antibodies: gamma, alpha, epsilon, mu and delta. The most abundant is IgG, which is transplacental, which can cross the placenta from maternal body and enter into fetal blood. Is that clear? We shall discuss about this bit later. Primary lymphoid organs and secondary lymphoid organs. What are these lymphoid organs? Primary lymphoid organs are the organs where the production and maturation of lymphocytes will take place. Whereas secondary lymphoid organs, even though they are not involved in either in the production or in maturation, they help in exposing these lymphocytes to the antigen. When the antibodies are exposed to the antigen, they will proliferate into effector cells. See here, I'm talking about our immune system. Human immune system, it consists of primary lymphoid organs, our immune system consists of primary lymphoid organs and secondary lymphoid organs. Primary lymphoid organs are the organs which helps in production and maturation of lymphocytes. Maturation of lymphocytes. What are lymphocytes? Lymphocytes are white blood cells. Leukocytes, yesterday we have discussion about white blood cells. There are 2 types of white blood cells, agranulocytes and granulocytes. Granulocytes include neutrophils, eosinophils, basophils, which contain enzyme-rich granules, whereas agranulocytes are monocytes and lymphocytes. The production of blood is known as hematopoiesis. Hematopoiesis will take place in red bone marrow. That means these lymphocytes are also produced in bone marrow. Leukopoiesis means production of white blood cells. We have discussed all these terms: leukopoiesis, hematopoiesis, erythropoiesis. Poiesis means production. So lymphocytes, these are produced in bone marrow. Both B lymphocytes and T lymphocytes are produced in the bone marrow. So the organs where production and maturation of lymphocytes takes place are known as primary lymphoid organs. Then what are secondary lymphoid organs? What comes under secondary lymphoid organs? We shall see. See, secondary lymphoid organs, even though they are not involved in the production and maturation of lymphocytes, they will provide site for interaction of lymphocytes with the antigen, which will make these lymphocytes to become effector cells. What is this effector cells? If you see what comes under this primary lymphoid organs here. Primary lymphoid organs include bone marrow and thymus. Bone marrow. In the bone marrow, production of both B lymphocytes and T lymphocytes will take place. Important. The production of T lymphocytes. What is the site of production of T lymphocytes? It is bone marrow only. But T lymphocytes will get matured in thymus. Is that clear? Maturation of T lymphocytes. Maturation of T lymphocytes will take place in thymus. But bone marrow, see this B lymphocytes, they will get produced and matured in bone marrow itself. Is that clear? So maturation and production will take place in the primary lymphoid organs. The primary lymphoid organs are bone marrow and thymus. Whereas secondary lymphoid organs are spleen, Peyer's patches, which is present in the small intestine, lymph nodes, and spleen, Peyer's patches, and lymph nodes, and even tonsils. These are all secondary lymphoid organs. Is that clear? See, when a B lymphocyte comes in contact with an antigen, it will produce two types of a cell population. What are they? Memory B cells and effector plasma cells. Hope you remember. See here, when a B lymphocyte is coming across with the antigen, so it will proliferate into two types of cells: effector plasma cells and memory cells. Isn't it? The production and maturation has taken place in the bone marrow. But these effector plasma cells, how they become effector plasma cells? When they are produced in the primary lymphoid organ, they will migrate from the primary lymphoid organ to secondary lymphoid organ to become these effector plasma cells. Hope you are following. Once again, the production and maturation of B lymphocytes has taken place in the primary lymphoid organ. In here, the B lymphocytes and T lymphocytes, after maturation, they come as, or they transformed as antigen sensitive lymphocytes. Antigen sensitive lymphocytes. Where? In the primary lymphoid organ. But where they are getting interacted with the antigen means in the secondary lymphoid organ. They will leave the primary lymphoid organ and migrate to these secondary lymphoid organs and get interacted with the antigen. Is that clear? So they will become, after coming to the secondary lymphoid organ, they will become effector cells, effector plasma cells. Is that clear? So primary lymphoid organs are bone marrow and thymus. Secondary lymphoid organs are spleen, Peyer's patches, lymph nodes, and tonsils. The secondary lymphoid organs provide site for interaction of lymphocyte with the antigen, by which the lymphocytes will become effector cells. Is that clear? No. So we know where bone marrow is located, in the long bones. In the bone marrow cavity, the production of these lymphocytes will take place. Production of white blood cells is known as leukopoiesis. And thymus. Coming to second one, that is thymus. Thymus, it is an endocrine gland. It is located, it is very small gland located near heart, below the breastbone. What is the breastbone called? Sternum. Sternum. Below the breastbone and above the heart it is located. The size of the thymus as the age of the person increases, as he is growing, the size of the thymus will be degenerated or size decreases. This progressive degeneration of the thymus gland, it is known as atrophy. Atrophy. What does atrophy? As the age increases, the size of this thymus gland decreases. That is why old age people are more vulnerable, are more susceptible to the diseases, means as there is no thymus gland, there is no maturation of T lymphocytes. What happens if there is no T lymphocytes? Fishes. They are the actual cells of our immune system. If one of these cells are not there, our immune system will become weak. So they are more susceptible to diseases. Is that clear? Thymus is a short endocrine gland which is located near the heart, below the sternum, and the size of the thymus will undergo degeneration by a process called atrophy. And that is the reason why old age people will be more susceptible to diseases, because the number of T lymphocytes will be reduced as the age increases. Is that clear? This is about primary lymphoid organs. Now, secondary lymphoid organs: spleen, Peyer's patches, lymph nodes and tonsils. Spleen. It is a large bean-shaped structure located near the duodenum, and we have studied spleen is a graveyard of RBC. Function importance here. The secondary lymphoid organs provide the site for interaction of lymphocytes with the antigen, with which the lymphocytes now become as effector cells. Important one. Spleen, it is a large bean-shaped structure located at the duodenum, near the duodenum. Okay, and spleen, it acts as a filter. Whatever the antigens coming through the blood, it will filters, and it is a big reservoir of RBCs, reservoir of erythrocytes, and spleen, it is commonly called as graveyard of RBC. Why it is called graveyard of RBC? The RBC lifespan is 120 days. After 120 days, the dead RBC or the damaged RBC will be stored and destroyed in this spleen. So it is a graveyard of RBC. It is filter for all blood-borne antigens. blood-borne antigen. It is acting as a filter for the blood-borne antigens. Is that clear? And second one, Peyer's patches. These are small patches which are present near the small intestine region, and they will also help against this fighting against the pathogens. Lymph nodes. What are all these lymph nodes? These lymph nodes are small swelling-like structure or small nodules which are present near. See here, lymph nodes. There are small nodule-like structure which are located near the forearms, forelimbs, in the shoulder region, apart from that, in the near the genital regions also, or in the groin region. Lymph nodes are located near the shoulder region or in the forelimbs region and in the groin region. These will trap the antigens. Immune responses are because of trapping the antigens near these lymph nodes. Lymph nodes are small nodule-like structures which are located in the shoulder region or in the lymphatic system, which can trap the antigens, and these antigens which are trapped in these lymph nodes are responsible for eliciting our immune responses. And tonsils, as we studied in the digestive system first year, these are also lymphoid structures which are located near the pharynx region. Okay, these are all secondary lymphoid organs which helps in interaction of lymphocytes with the antibody. Important: first primary lymphoid organs and secondary lymphoid organs. See here, two types of immunities are there with response to B lymphocytes and T lymphocytes. The immunity mediated by B lymphocytes which is mediated by B lymphocytes is known as humoral immunity. Humoral immunity. And the immunity which is mediated by T lymphocytes is known as cell-mediated immunity. How the mechanism? A bit later. See, what is humoral immunity? In the starting classes of immune system, human health and diseases, we have talked about good human hypothesis. Have you remember? Humor means what? Body fluids. Killing of antigen is mediated through the body fluids by the production of antibody. So it is known as humoral immunity or antibody mediated immunity. Humoral immunity or antibody mediated immunity. Why it is called as humoral means the fighting of antibody with the antigen is taking place through the body fluids, through blood and lymph. So it is known as humoral immunity. And why it is known as antibody mediated immunity? Because when the B lymphocytes proliferate, they proliferate into plasma B cells and memory cells. These plasma cells will produce the antibodies and fight against with antigen. So it is known as fight against with the antigen. So it is known as antibody mediated immunity. These T lymphocytes, as they do not have the capacity to produce antibodies, it will kill the antigen by through the different types of cells like macrophages. How it is known as? This is known as cell mediated immunity. When we look at this proliferation, see here, B lymphocytes are of two types. B lymphocytes, many times I have written, see here. B lymphocytes are of two types. They are plasma cells and memory cells. Whereas T lymphocytes are of four types. They are T helper cells or helper T cells. We can write like this: T helper cells or CD4 cells. Cluster differentiation 4. They will have a surface marker. That surface marker it is cluster differentiation 4 it is. And TC cells. TC means cytotoxic T cells, which are also known as killer T cells. Cytotoxic T cells also known as killer T cells. And next one, suppressor T cells. Suppressor T cells. And fourth one, T cells. Memory T cells. So cell immunity and the cells are produced by B lymphocytes is known as are of two types. They are plasma cells and memory cells. The immunity mediated by T lymphocytes is known as the cell mediated immunity, and there are four different types of T lymphocytes: helper T cells, cytotoxic T cells, suppressor T cells and memory T cells. Helper T cells, cytotoxic T cells, suppressor T cells and memory T cells. The actual cell which helps in production of antibody, which is known as antibody factory, is plasma B cell. Plasma B cell, which can able to produce antibodies. So, expected questions here. Which cells are known as antibody factories? Among all these cells, which cells are known as antibody factories? Means plasma B cells are known as antibody factories. Antibody factories. Is that clear? Now, memory cells. What is the function of memory cells? If the same pathogen is encountering a person for the second time, that means the subsequent encounter of a pathogen to a person, then what happens? Then these memory cells will recognize that antigen and immediately kill that antigen and will not allow that antigen to cause disease to the person for the second time. We shall see what is primary and secondary responses, then we can understand. What does these memory cells? As the name indicates, these memory cells will store the information regarding the antigen, and when the same antigen, the subsequent encounter of the antigen, in that subsequent encounter, these memory cells will recognize the antigen very quickly and they will destroy that antigen immediately, and it will not allow the person to fall ill for the second time. And T lymphocytes, see here, helper T cells. As these T lymphocytes do not have the capacity to produce the antibodies, they will help, these T helper cells will help these B lymphocytes in the production of antibodies. Apart from that, it will also interact with the macrophages in killing the antigen. And cytotoxic T cells. These cytotoxic T cells will produce an enzyme called perforins. they will produce an enzyme called perforins. These perforins will cause holes in the structure of the antigen and completely will destroy the antigen. So as they are killing the antigen by creating certain holes in the structure, by secreting certain chemicals called perforins, they are called as killer T cells or cytotoxic T cells. What is the toxic substance it is secreting? Perforins. And suppressor T cells. These suppressor T cells will suppress the cancerous cells or any cell which is acting against our own immune system. Is that clear? Suppressor T cells will suppress any cell which is acting against our immune system. And memory cells, as the name indicates, they will recognize the antigen in the subsequent encounter. So these two are lymphocytes, T lymphocytes and B lymphocytes. Now, see here, primary immune response and secondary immune response. Immune response. Immunity. What is immunity? The ability of our body to fight against with the disease-causing pathogen. When a person is exposed to a particular antigen for the very first time, how our immune system is acting, the first response of our immune system is known as primary response, and the subsequent encounter of the antigen and how our immune system is acting against that antigen, it is known as secondary immune response. So immune responses are of two types: primary response and secondary response. Primary response and secondary response. See here. If a person is affected with certain virus for the first time, then our immune system will take characteristic features of the primary response is that primary response is slow. It should recognize that antigen and it should react with that antigen. It should form these antibodies and kill that antibody. So it will take time for our immune system to destroy that antigen. So it is a slow process. Primary response is slow in its mechanism and it is with less intensity. Less intensity means less effective. Okay. Whereas in the subsequent encounter, whereas see here, the subsequent encounter of a pathogen to the person, then how the secondary response will be. The secondary response is very quick or fast and is very quick or fast and with high intensity, very effective it is with high intensity. The intensity of the response is very high. Immediately it will recognize and they will kill the pathogen. So such type of it is known as anamnestic response. The anamnestic response of our immune system for the subsequent encounter of the antigen is known as secondary response. Once again, see the definition of secondary immune response. The anamnestic response of our immune system to the subsequent encounter of the pathogen is known as secondary immune response, which is characterized by a quick response and highly intensified response in killing the pathogen. How the secondary response is very quick and how it is very high intensity means because of presence of these memory cells. These memory cells will recognize the antigen very quickly and they will stimulate these plasma cells to secrete these antibodies and destroy the antigen immediately. But whereas in the primary response, the B lymphocytes, they should get proliferated into plasma cells and they have to prepare these antibodies. So for the recognition and interacting with the antigen, it will take time and it will be less effective in case of primary immune responses. Is that clear? So primary immune response primary immune response is slow and less intensive, whereas secondary immune response is quick with high intensity. Is that clear? Is that clear? Now, our immune system, types of immunity. Broadly, immunity is of two types: innate immunity and adaptive immunity. Innate immunity, already we have discussed, the immunity which is present right from the birth and which is non-specific, it is known as innate immunity. Whereas adaptive immunity, the immunity which is acquired after exposing to the pathogen, is known as acquired immunity. Immunity is of two types: innate immunity and acquired immunity, or adaptive immunity. Acquired immunity or adaptive immunity. Innate immunity, the immunity present in an individual right from his birth, is known as innate immunity. Since birth. Okay, which is non-specific. That means what comes under non-specific? First line of defense and second line of defense, it comes under non-specific and innate immunity. Whereas acquired or adaptive immunity, which includes third line of defense, that means after exposure to the pathogen, the immunity acquired by a person after exposing or after interacting with antigen or a pathogen. Is that clear? See here, immunity which is present right from the birth is innate. Our immune system is not involved in providing immunity against the pathogen. That means our body is provided with ready-made antibodies or processed antibodies in case of passive immunity. Our T lymphocytes and B lymphocytes has no role in destroying the pathogen in case of passive immunity. Why? Because the antibodies are directly injected into the person in case of passive immunity. Once again see here, acquired immunity is the immunity which is attained by an individual after exposing to the antigen. It is of two types, passive and active. Passive immunity, the immunity gained by the person by taking the processed antibodies directly. Is that clear? That means our B lymphocytes are not involved in the production of antibody and going that antibodies to kill that antigen. We have taken the antibodies from an external source. So it is known as passive immunity. Hope I am clear. See here, and whereas active immunity. Active immunity means our own immune system has involved in the production of antibodies and killing the antigens. When our own immune system is involved in fighting against the disease causing pathogen, then it is called as active immunity. See here, again passive and active are divided into naturally acquired and artificially acquired. Passive active and naturally acquired active, artificially acquired active immunities. What are all this? Shells. Hope I am clear. In case of passive immunity, we are taking the ready-made antibodies or processed antibodies from the external source. Our immune system is not involved in the production of antibodies in case of passive immunity. Whereas in case of active immunity, our own immune system is fighting against with the disease causing pathogen, that is active immunity. That means who are producing antibodies in case of active immunity? Our own B lymphocytes are involved in the production of antibodies. Again passive and active are again they are divided of antibodies. Again passive and active are again they are divided into naturally acquired or artificially acquired. See, immunity first broadly it is classified into innate and adaptive. Innate means the one which is present right from the birth and it is non-specific. Now I am talking about acquired immunity or adaptive immunity. Acquired immunity, see here. Two broad classifications, no? Passive and active. Passive and active. Passive. Passive is again divided into, it is of again two types. Naturally acquired passive immunity. Naturally acquired passive immunity and artificially acquired passive immunity, okay, and active immunity. Active immunity is also naturally acquired active immunity and artificially acquired active immunity. Artificially acquired active immunity. How we are going to understand these immunities? Naturally acquired passive immunity. Say for example in case of infants. In case of infants they still not developed their immune system completely. They cannot fight with the antigen. So what happens? These infants are provided with ready-made antibodies. Who is providing? Mother provides antibodies through the placenta in case of fetal stage, and in case of young ones through the breastfeeding. See, IgG antibodies, immunoglobulin G antibodies, they are transplacental antibodies which can cross the placenta, and they can take this the IgG can cross the mother's placenta and enter into the mother, is known as naturally acquired passive immunity. Is that clear? And IgA antibodies which are present in colostrum. Mother's milk contains mother's milk, the very first secretion of mother's breastfeeding, it is known as colostrum. It is rich in antibodies, that is IgA antibodies. And the immunity gained by the fetus because of these antibodies through the mother, it is known as naturally acquired passive immunity. Where these antibodies has produced? The antibodies is produced in the mother. But who is gaining immunity? The fetus or the young one or the infant is gaining immunity. That means the fetus is getting immunity not by his or her immune system. The processed antibodies or the ready-made antibodies are provided to the fetus or provided to the infant to gain the immunity. So such type of immunity where antibodies are not produced in their own immune system but they have taken from an external source, such type of immunity is known as naturally acquired passive immunity. Is that clear? Now, artificially acquired passive immunity. See, a person got tetanus, or a person is seriously injured because of a snake bite. Seriously affected because of a snake bite. In such condition, in order to save the life of an individual, processed antibodies or ready-made antibodies are injected to the person. Here, snake bite, what the snakes will inject? Venom. Venom is injected into that person. So the person immune system has no time in recognition of this venom or toxin and activating the B lymphocytes for the production of antibodies. There is no such long time in order to save the life of that person. So at that instant, the person who is affected is injected with processed antigens. Processed antigens. Okay. Where we will get these processed antigens? We shall see these processed antigens with see if a heat killed or attenuated antigen is processed antibodies. How we will get these ready-made injections or anti-venom? How we will prepare anti-venom? Means attenuated antigens or heat-killed or inactivated antigens are injected into another animal, and it will be sensitized and it will produce the antibodies. And if we collect the blood sample and separate the serum, in that serum we will contain these antibodies. Okay, so that when the antibodies are injected to the person at that instant to save the life of that person, okay, that means processed. And here the injured person's immune system is not involved in the production of antibodies. We have taken the antibodies from an external source, say for example horse or mouse or some other animal. From them we have taken the antibodies. If our immune system is not involved in the production of antibodies, then even though then we got the immunity, then such type of immunity is known as passive immunity. Here it is natural. Why? Because through the mother to fetus it is a natural instant. Whereas here it is artificial because a person when he got affected with deadly diseases, then the antibodies are given to the ready-made antibodies are given to that person. So it is artificially acquired passive immunity. Is that clear? Now, naturally acquired active immunity. So what I said, active immunity means who is involved? Our own immune system, T lymphocytes and B lymphocytes are involved in the production of antibodies and in destroying the pathogen. So naturally acquired active immunity means, say for example, a person got affected with smallpox. The person who is affected with the smallpox for first time, and there is, if the same smallpox virus has affected that person, for the subsequent encounter he will not get the disease. In majority of the cases, second occurrence of smallpox will not, we cannot see, because already our immune system has recognized the antigen which is causing the smallpox, and it has proliferated into two cells, plasma cells and memory cells. These memory cells has recognized the antigen, they have the memory about the antigen, and in the subsequent encounter they will kill the antigen, so the person will not come across with the disease for the second time. So such type of immunity in natural infections, it is known as naturally acquired active immunity. This active immunity, it is because of memory cells. When memory cells are present in the subsequent encounter, the person cannot fall into disease. Hope I am clear. Once again, smallpox. Here, when a person got smallpox, our immune system will recognize that antigen, it will fight against with that antigen, and the antibodies are formed, and these antibodies will kill that antigen. And when the B lymphocytes, they have proliferated, these memory cells will make a record of that, and they will fight against with that antigen in the subsequent encounter. Such type of immunity acquired by the person is known as naturally acquired active immunity. Now, artificially acquired active immunity. What is this artificially acquired active immunity? The immunity we gain by vaccination. The immunity we gain through vaccination is known as artificially acquired active immunity. See what happens during vaccination. During vaccination, a heat-killed or weakened or attenuated antigens will be deliberately introduced into our body. Once again, so what is the phenomenon behind this vaccination means, or weakened means what is antigen? The antigen which can cause the disease, but the disease causing ability of that antigen will be nullified in case of vaccination. The same antigen is taken, but the disease causing ability of that antigen will be nullified or neutralized. Such type of antigens are known as heat killed, or they will be kept in formalin, so they are known as weakened or attenuated antigens. When these attenuated antigens are deliberately introduced into our body, what happens? Our immune system will recognize those antigens. What our immune system will do whenever it will come across with the antigen? Will it keep quiet? The B lymphocytes will recognize that antigen and immediately produce antibodies. Is that clear? Then, what happens if, when they are introduced into our body, antibodies are formed? Antibodies are formed. Then, that means to us, already we have come across with it, which are already formed, they will identify that antigen and kill that antigens. So the person will not fall into disease. That is, such a type of immunity gained by the person by taking these attenuated antigens is known as artificially acquired active immunity. So vaccination comes under artificially acquired active immunity. Why it is active immunity? Because vaccination does not give the antibodies directly. Vaccination does not give the antibodies directly. In vaccination, what are injected into our body? A heat-killed pathogen or an attenuated pathogen or a weakened pathogen is introduced to our body. Antibodies are not introduced into our body in case of vaccination. When the antibodies are given, antibodies are directly given when the person is seriously ill. Okay, that comes under passive immunity. But whereas vaccination comes under active immunity because antibodies are not given and heated or attenuated antigen is given. So what happens? Our immune system, our own immune system will recognize those antigens and produce the antibodies against that antigen. If our immune system is involved in the production of antibodies, then such type of immunity is known as active immunity. Is that clear? But it is not natural. We are taking purposefully, we are taking the vaccine, so it is artificially acquired active immunity. Hope I am clear with the immunity. A brief recap here. Acquired immunity is of two different types, passive immunity and active immunity. Passive immunity, there is no role in the production of antibodies for our immune system. We are taking the antibodies from an external source in case of passive immunity. Whereas in case of active immunity, our own immune system is involved in the production of antibodies. That is active immunity. Again, passive immunity is naturally acquired passive immunity and artificially acquired passive immunity. In case of infants, the immunity gained by the infant or the fetus by acquiring the antibodies from the mother, such type of immunity is known as naturally acquired passive immunity. Matching very important on this thing. And artificially acquired passive immunity. A person who is seriously affected by a dreadful disease, in such condition the antibodies, processed or ready-made antibodies are injected to that person. So that comes under this artificially acquired passive immunity. Now, naturally acquired active immunity. That means through the natural infection. Say for example, nowadays in this COVID situation, if a person has affected with coronavirus, now there is no vaccination against this corona. The person immune system is self-involved in fighting against with the coronavirus and the antibodies are produced in their body. So for the second time, if the same virus is infecting the person, he will not fall into the disease because their memory cells are activated and they will produce the antibodies. That's why nowadays they are using the serum therapy or serum technique, plasma transfer they are using. What does that plasma contain? That plasma contains the antibodies. Such a type of interaction of a disease, such type of interaction of our immune system to the disease causing pathogen, where itself it is involved in the production of antibodies, is known as naturally acquired active immunity. Is that clear? Now, artificially acquired active immunity, that is through vaccination. Before the actual infection, we are precautionarily taking the heat-killed or attenuated antigens by which our immune system will recognize those antigens and the B lymphocytes will produce the antibodies against that particular antigen. And if the same type of antigen comes into our body, then these antibodies will fight against that disease and we will not fall into disease. That it is known as artificially acquired active immunity. Still more regarding this vaccination, we shall see. That means Edward Jenner has identified this phenomenon of vaccination. What he has given the statements means initial exposure of a mild antigen will give protection against the virulent pathogen. Once again, see the statement. Edward Jenner has stated that initial exposure to the mild pathogen will give or will provide immunity against the exposure to the virulent or a strong pathogen. That's what it is in the vaccination. What happened? We are exposing to a weakened pathogen. When a virulent pathogen or a strong pathogen comes to our body, then the antibodies which are formed already, they will fight against with a strong form of antigen. I hope I am clear. I'll write the statement regarding this. Edward Jenner. See, regarding vaccination. See, regarding vaccination, what happened is Edward Jenner, what he has identified. First, he has what he has see here. When a person or the cow maids, those who are doing milking, when they got affected with the cowpox, they are not affected with smallpox. Then what he has identified means he has taken the antigen which is causing cowpox and injected to a person, and he has after later when the disease has resided to the same person, he has injected a pathogen which is causing smallpox, but the person has not fallen to ill. Then what he has stated that Edward Jenner has stated that initial encounter to a attenuated form of antigen will provide immunity against strong or virulent form of antigen. This is the theme behind vaccination. See, in vaccination, we are not provided with antibodies but with antigen. That means when we are initially encountered to the attenuated pathogen, in the subsequent encounter, it will provide immunity against the stronger virulent form of antigen. That is vaccination. So we are done with immune systems, and what are remaining means allergic reactions and HIV and drugs and adolescents. Before going to that, see here a brief recap of these two classes. See here, immunity. We have studied three lines of defenses: first line, second line, and third line, or non-specific immunity and specific immunity. Non-specific immunity, it is innate immunity right from the birth, and it is generalized response of our body to any kind of pathogen. Whereas third line of defense or specific immunity is highly specific to the pathogen. Apart from that, we have studied about what are all the lymphoid organs, primary lymphoid organs and secondary lymphoid organs. How our immune responses are there, primary response and secondary response, and immunity, how many types of immunity is there, innate immunity and acquired immunity. Again, acquired immunity is again passive and active. These are all we have studied. And remaining allergy. We shall talk about allergy. What is allergy? The exaggerated response of immune system is known as allergy. We can term this allergy or as hypersensitivity. What is hypersensitivity? The exaggerated immune response of our body for the subsequent encounter of a pathogen is known as hypersensitivity or allergy. Why it is subsequent encounter? Because in the first encounter, our immune system will become sensitive to that pathogen. Once again, the exaggerated immune response of our exaggerated immune response encounter of a pathogen is known as allergy or hypersensitivity. In the first interaction, our immune system will become sensitive to the particular antigen, and the agent which is causing allergy is known as allergen. What is allergen? The agent which cause allergy. The agent which cause allergy is known as allergen. What are those allergens? Smoke and dust particles. Yes, if we work in the dust means we will come across with these such allergic reactions. Dust, smoke particles. Most of the people are allergic to pollen grains and certain spores of the plants. Pollen grains or spores of the plants. And even animal dander. What is this animal dander? Means hair of the dog or cats. Okay, their skin flakes from the skin of the animal, that is known as animal dander. All these which are causing an exaggerated immune response, they are known as allergens. What is allergy? An exaggerated immune response to the subsequent encounter. The subsequent encounter of pathogen. The exaggerated immune response to the subsequent encounter of the pathogen or allergen is known as allergy or hypersensitivity. What are all the symptoms of these hypersensitivity or allergy? That means, see, if a person come across with these pollen grains, or some people are allergic even to certain food components also, or so they are allergic to certain chemicals. Say for example, penicillin injection, it is not susceptible to all. It is not reliable to all. So it is allergic to some people. So before giving that, a test should be done to such patients who are allergic to certain drugs. Okay, what are all the symptoms? What are all the symptoms we can observe during allergy? See here, the very first symptom, sneezing, watery eyes, running nose, or in case of these pollen grains or dust, in case of asthma patients, it is difficulty in breathing. Difficulty in breathing. All these are the symptoms, or sometimes turning to redness. Redness. All these are the symptoms of allergy. Why all these are? What chemicals or what immune responses are responsible for the symptoms of allergy are? In case of allergic reactions, IgE antibodies are produced. IgE antibodies are produced in case of allergic reactions. They are fighting against with these allergic antigens or allergens. Apart from that, during allergic reactions, number of eosinophils increase. What are these eosinophils? Eosinophils are granulocytes, leukocytes, white blood cells. They are also known as acidophils because they are stained with acidic dyes. As the number of eosinophils increase, the condition which is known as eosinophilia or eosinophilia. Eosinophilia. Is it clear? During allergic reactions, what are the antibodies are found in the blood? IgE antibodies are produced, and the cells which are fighting against with these allergens are eosinophils. Their number increases. Abnormal increase in the number of eosinophils. The condition is known as eosinophilia or eosinophilia. And what chemicals are responsible for these allergic reactions? Means what cells means? Mostly mast cells and basophil cells. Mast cells and basophil cells. Mast cells and basophil cells will secrete certain chemicals like histamine and serotonin. Histamine and serotonin. What they will do? Histamine, it is a vasodilator. What does vasodilator do? The diameter of the blood vessel increases. When the diameter of the blood vessel increases, capillary permeability increases. When the capillary permeability increases, loss of fluids from the blood will take place, which will lead to inflammation or which will lead to this redness or other symptoms of this allergy. See here. Histamine and serotonin, which are produced in response to allergy, this is the diameter of the blood vessel. The diameter of blood vessel increases, and the blood vessel, it is lined by this squamous epithelium. These are endothelial cells of the blood vessel. Now, when the diameter increases, the space between these endothelial cells increase. So what happens? The blood by flowing here, the permeability of the blood capillaries increases. So the loss of fluids will take place. Loss of fluids. It will lead to swelling or this redness will occur. The flow of blood also increases in case of these allergic reactions. Histamine and serotonin, they will cause vasodilation. They will cause vasodilation. What does vasodilation means? Vasodilation means increase in the diameter of the blood vessel. When the diameter of the blood vessel increases, the blood capillary permeability increases. Capillary permeability increases. When capillary permeability increases, then loss of fluids from the blood vessels will take place, will cause allergy. Then what should be given? Anti-allergens should be injected to the person who is suffering from allergy. Anti-allergens or adrenaline or epinephrine. Adrenaline should be given to the person. This adrenaline is a vasoconstrictor. which is acting against this histamine. Okay, who is secreting this adrenaline? Adrenal medulla, catecholamines, adrenaline or epinephrine. Hope you have remembered first year topic. Okay, this epinephrine will act as an anti-allergen and will suppress the allergic reactions. Most of the people who are living in the metropolitan cities are more susceptible to these allergy because there is reduced immunity for them, as they are more exposed to these dust particles and all. They are becoming very sensitive to these allergens. So because of this sensitivity, most people are falling into these asthma or to these allergic reactions only because of increased sensitivity of the person and decreased immune power of an individual. Mostly those people who are living in metropolitan cities. Okay. So once again, a brief recap of allergy. Allergy means the exaggerated immune response of our body for the subsequent encounter of a pathogen is known as allergy. Okay. What is the agent which is causing allergy is known as allergen. What are the antibodies that are produced in response to allergy is IgG, epsilon. And next, what the cells are increased means eosinophils are increased. What are all the symptoms of allergy? It includes sneezing, watery eyes, running nose and difficulty in breathing, mostly in case of asthma. And apart from that, which cells are responsible for these allergic reactions are mast cells and basophil cells, which are secreting the chemicals like histamine and serotonin, which are vasodilators. What happens when the vasodilation occurs? The diameter of the blood vessel increases. As the diameter of the blood vessel increases, capillary permeability increases. When the capillary permeability increases, there will be loss of fluids from the blood, which will lead to certain allergic reactions. So, in order to suppress these allergic reactions, anti-allergens should be given like adrenaline, which is against to this or which is antagonistic to histamine. Then what happens? This will suppress the allergic reactions. In the next class, we shall study about HIV and even drugs and adolescents. Thank you. Amma.